Hello, good morning, good evening to everyone joining with us today from around the world to the 11th episode of PhD Talks by NcodeX. So this month, July 2022, is a special month for us at NcodeX. We're turning 1 year old this month. So last year, 2021, July, we officially relaunched the Young Research Initiative as the company NcodeX, and since then we have worked with researchers from all around the world in research development and training young minds. So mentioning briefly who we are, the NcodeX's vision is to become the pioneer in interdisciplinary STEM, science, technology, engineering, and mathematics R&D innovation for a sustainable future by bridging the long prevailing gap between academia and industry. So how do we achieve our vision? By innovative research development and execution, and STEM coaching and training for young researchers to meet the global demand. So we gather early career researchers, senior scientists, and professionals from both academia and industry around the world into one roundtable to brainstorm ideas, share knowledge and experiences and expertise to collectively produce innovative, trending, global scale research and provide sustainable, innovative solutions to meet the global challenges. So to know more about NcodeX research and our available training programs, please visit www.encodexpert.com, which is also given in the chat box here. So today, for our 11th episode of PhD Talks by NcodeX, we're joining with my friend, my past colleague, Dr. Nirusha Weerasinghe, all the way from London. So welcome, Nirusha. Good to have you here with us today.
Good morning. How are you?
Good, thank you. So Dr. Nirusha Weerasinghe is a registered scientist and a chartered biologist, currently working as an associate practitioner at St. Thomas's Hospital, London. Nirusha obtained his BSc Honours in Biomedical Sciences from Northumbria University of Newcastle, UK. His Master's in Biomedical Sciences, specialised in Cellular Pathology from Middlesex University, UK. And he recently completed his PhD in Biomedical Sciences, emphasizing in Oncology from Middlesex University, UK. So today he will be talking to you about vasculogenic mimicry in bladder cancer, an interesting topic for all future cancer researchers, I believe. For any questions and discussion points, please post them in the chat box or in comments. We will discuss them after the presentation during the Q&A session. So without further ado, Nirusha, over to you.
All right, thank you. All right, good morning and good afternoon and good evening for all around the world, people. So I'm going to give you a brief summary of my PhD research, which was on vasculogenic mimicry in bladder cancer. And then I will also give you some tips for PhD students, how to make sure that your PhD is a success and then get the best out of your PhD time in your university. So bladder cancer, as you all know, it's an abnormal growth of tissue around the bladder lining. And there are 3 types of bladder cancer tissue, such as transitional cell carcinoma, squamous cell carcinoma, and adenocarcinoma. This depends on how much the cancer grows into the tissue. As you can see in the picture below, there are 4 layers of the bladder lining. So if the cancer grows in the superficial layer with the transitional epithelium, we call it transitional cell carcinoma. And if it moves towards the muscle layer, it becomes muscle invasive. And if it moves further down, it becomes metastasized. So there are 2 main types of bladder cancer depending on the invasiveness. One is non-muscle invasive bladder cancer, and the second one is muscle invasive bladder cancer. So as you can see in this image, superficial bladder cancer occurs in the lining of the bladder, and if it moves towards the muscle layer, it becomes muscle invasive, and if it moves further down, it becomes metastasized and spread around the bladder and moves towards other parts of the body as well. So as you know, cancer cells require nutrients and excretion of waste material. So this is due to a process called angiogenesis, where formation of blood vessels. So a lot of researchers have found anti-angiogenic therapy to inhibit the process. So inhibiting the process, they can possibly reduce the growth of the cancer cells. But however, some of them have seen a resistance to this anti-angiogenic therapy because some of the cells survived and they started growing back again. And they come up with this theory called vascular mimicry, where these tumor cells make their own blood vessels. They line around and make their own blood vessels. So this is called vascular mimicry. As you can see in this picture, a normal blood vessel is aligned by endothelial cells, while a vasculogenic mimicry channel is aligned by cancer cells. So I did a double staining technique, immunohistochemistry on bladder cancer tissues, where I stained with an endothelial marker and a periodic acid shift staining. So when you do distinguish a bladder cancer vascular mimicry channel with a blood vessel, you don't see the endothelial marker in the vascular mimicry channel. So as you can see, the arrows that I have shown in red shows the presence of pass staining, but negative staining of endothelial marker, which suggests that these are vascular mimicry channels. If you look at figure E, the brown staining, which is the endothelial marker, suggests the presence of blood vessels. These are indicated in yellow arrows. The red arrows shows the presence of vascular mimicry channels. And if you look around it, you can see the presence of cancer cells as well, because these are metastasized areas of the cancer. And you can see the presence of red blood cells within these vascular channels, which is also another theory that was formed, that vascular mimicry channels carry red blood cells because they bind to the host blood vessels. Therefore, to see this in a three-dimensional structure, I was able to create a 3D model. So what are the benefits of 3D model? You can see the 3D structure of a cancer, and then it will preserve the morphology and the divisions of cells. Also, you would be able to see the cell-to-cell interactions and extracellular matrix networks that with appropriate treatment. Also, you will be able to see the angiogenesis of these tumors. So this was one of the main areas of my PhD, which I made a 3D model. So I used bladder cancer cell lines T24 and SCABR to make these 3D models. So what I did, I made an extracellular matrix using collagen and fibronectin, which are ECMs. And then on top of that, I cultured cells and then left it over for them to grow. As you can see, we were able to see vascular channels in these 3D models, sorry. But how can we distinguish whether it's a blood vessel or is it a vascular mimicry channel? So I was able to do a double staining with same as the histochemistry, as an endothelial marker with laminate. Laminate is also a basement membrane marker. So laminate should be positive and endothelial marker should be negative if it's a vascular mimicry channel. As you can see, these are negative for endothelial marker, but positive for laminate, which suggests that these are vascular mimicry channels. Overall, in conclusion, during my PhD, I was able to exhibit the presence of vascular mimicry channels in bladder cancer, and I was able to confirm a theory that vascular mimicry channels were observed in high grade and high stages of bladder cancer, and it can be used as a potential prognostic marker. Also, I was able to find out that there were several signaling pathways, such as TGF-beta signaling pathway, Wnt signaling pathway, even VEGF pathway involved in this process. Also, different factors were involved as well, such as cancer stem cells, because cancer cells exhibit the phenotype of endothelial cells when they're forming vascular mimicry channel. So this can be due to the presence of cancer stem cells. So cancer stem cells is one of a major factor that's involved in vascular mimicry. Now, I would like to give some tips for all of you if you're doing research or if you're planning to do research. These are some of the tips I would love to give you, because I have gone through the whole process, so I know what is good and bad in research. So if you talk to any PhD student, there are several struggles. So the first one is isolation. For an example, if you come to another country to do a PhD, you will be missing your family and friends. That's very common. So it's one of the major problems for a PhD student, isolation. Second one is stress, the workload. Because if you're a PhD student, you have to do things on your own. You have to plan things. You have to work on how to do things better. And for an example, if you're a science student like me, if you do a certain practical 100 times, you'll get only like 1 or 2 good results. So that is also stress. And if you come up with an idea but your supervisor doesn't agree with it, then you'll have a conflict with your supervisor. So this is also another major issue. The most important issue is funding. So if you're not a student who's not fully funded, funding is a big issue, because you need to buy certain things for your PhD. And then if you don't have enough funding, it will be a major issue. Time management. You need to time manage really well, unless it will be a failure. So you need to make sure that how you're going to manage the time and make use of the time perfectly. Also, work-life balance. I have seen a couple of my friends leaving PhD because they couldn't balance work and life. It is fundamental, because your life apart from PhD is important as your PhD. Also, lack of support. You won't get enough support because it's your research. It's you who are going to do it. It's you who's going to work on it. So you won't get support from everyone. If your supervisor is nice, they will help you, but it's all on you. And then future concerns. It's also another major one, because people, once they do PhD, they just do it as a PhD, but they don't know what they're going to do in the future. So if you don't have a right plan of what you're going to do next, it will be another major issue. And another one is motivation. Motivation is a big problem with PhD students. You need to be motivated to do PhD, otherwise you will fall away. So what now? So the first advice I would love to give you is make a roadmap. So make a Gantt chart. Gantt chart is like a plan how we are going to do things in certain months or certain days. Okay, so if you plan from the first year, how you're going to plan till the last day of your viva, then it shouldn't be exact. But if you have a rough plan how you're going to do things, that will help you. And always create a plan of action, because you never know when your plan is going to work and when it's not going to doesn't work. So for an example, if someone falls sick, you won't be able to do certain things that you have already planned. So always have a backup plan. And the third one is to make sure that you have figures and legends and your methods written somewhere that you can remember. So for an example, if you do some certain experiment in your first year, and then you're using that result in your thesis, at the end of the thesis, you will not remember what you did. So make sure you write everything, note it down, make sure you write the methods as well, because we are all humans, we will forget things. Always take scheduled breaks. So don't work constantly on your PhD 24 hours. Take a break. Every 2 hours, every 3 hours, take a break. Just relax your mind, come back again. Because if you work constantly on it, it will affect you. Also, avoid distractions. When you're working on something, make sure that you're fully focused on that. If this is not only for PhD, this is for everything. So make sure that you are not distracted by someone or something else. So make sure that you give 100% to it. Make sure you do it correctly. And then stay motivated. Motivation is very key. So attend to seminars, attend to lectures, Go and meet people around, talk to them, stay motivated, because when you see someone doing something amazing, you feel like, oh, I'm doing something new as well, so I'm amazing too. So you keep yourself motivated. If you're not motivated, you can't do your PhD. Also have periodic reviews. So for an example, if you did something during a period of time, and make sure that that is approved and okayed by your supervisor, because you don't want to go back and do that again and again. But if you know what you have done is correct, show it to them, make sure they say, okay, this is right. If not, get their feedback, how you can do it better. That's very important as well. And always use appropriate referencing software, because as you all know, with PhD, you'll have hundreds and hundreds of references, but if you keep it at the end, you'll forget the references that you did. Then you have to research again and then put them back. But if you use a good referencing software, like Mendeley, for an example, sorry, it will store all the references that you have used throughout your PhD. And at the end of your thesis, you can put it as a bibliography or a reference list. So finally, I would love to tell you that never give up. Things may be hard, but if you quit, they'll never get better. Thank you very much.
Thank you very much, Nirusha. That was amazing talk. And I think I agree with everything you just said. And as a PhD student, I could use a reminder myself. So thank you very much for that.
Right.
Yeah, so let's talk a little bit about your PhD project. So what is the final outcomes that will contribute to sustainable future, and what are the key take-home messages from your research?
I would say that the finding of vascular mimicry in bladder cancer, it's a prognostic marker. So we have found different factors that are involved and signaling pathways. So I wasn't able to do all the signaling pathways because I didn't have enough time to complete it, because there's millions of signaling pathways involved. But the take-home message is we have found a different strategy to stop the progression of bladder cancer cells. So for an example, if we can use anti-angiogenic therapy, several cells will survive. But if you use anti-VM therapy, that would mostly halt most of the progression. So I think that's the main sustainability part of my PhD. And then it can be used for prognosis, diagnosis of bladder cancer patients, because currently the main diagnostic way is cytology. So this can be a good way of prognosing as well as diagnosing. So prognostic way as well, it's very important because there's not much prognostic markers for bladder cancer.
Perfect. So if someone continues in the same area as your research, is there any way we can incorporate other STEM disciplines like technology, engineering, and mathematics to bring the final outcomes to the communities in need? What is your approach in that?
Yeah, definitely, because it's not only about medical sciences. There's like mathematics involved, and computer sciences because not only in my research in general, if you take research, you need devices to create. You need biomedical engineering. You need computer sciences to create those coding for these technological devices. And then if you look at what else, mathematics, you need those mathematicians to do all the calculations for it. So science is not just only one. It is a cooperation of engineering, mathematics, and technology. So not only medical research, like I think every research management, even arts, you need a cooperation of everything. Interdisciplinary, it's very important.
Absolutely. Yes, so if you have any questions, by all means, unmute yourself and ask away. And if not, you can always post them in comments or chat, and then we'll direct the questions. Meantime, let's move on to the important part we talked about, doing a research. That was very important. And so at NcodeX, we always promote innovation, keep innovating and improving the rate of innovation over time. So what is your idea about the innovation and how much we need to think of it?
Innovation is very key, because if you're not innovative enough, you can't do research, because PhD research is not doing someone else's research. You have to be innovative to make sure that you do a research that is new. Sometimes there are things like, okay, if you start on a certain project and then someone else have done it recently, and then you should be innovative enough to tweak it a bit. So for an example, it happened to me. So I was working on actually cervical cancer. And then I saw a paper published like 2 months after I started my PhD on cervical cancer. So I changed my cervical cancer into bladder cancer. So you should be able to work a bit, because things are not the same every day, because there are millions and millions of researchers around the world doing different things. It can be your same topic as you as well. So if you are not innovative enough to change the topic a bit and change along the way, it's going to be hard. So you have to be innovative all the time.
I couldn't agree more. So there comes the resilience and flexibility.
Correct.
Yes, and also sharing and publications. They all come together with the innovation, I'm guessing.
Yeah, definitely.
Okay, so do we have any questions in the chat? No questions. So, okay, Nirusha. So before we wrap up, I know you talked a lot about how to do a research and good points to future researchers. So let me ask you like this. If you could go back to your few years back and give one advice to your younger self, what would it be?
Write your thesis along in the way. This is what I didn't do. So during my second year, I was focusing on my results rather than writing anything. So when I went to my third year, I forgot some of the methods. I'm sad to say, but I forgot some of the methods. But when I looked at the protocols and what I have done, luckily I have written them down on a book. So I use that as my like a refreshing method. But if you write your thesis along your way, because you can write your introduction, you can write your methods. You don't have to write your results or discussion yet. But if you can write your introduction and your methods, it will be easy for you. It doesn't have to be perfect. Just write it the way you want to write. Because in PhD, there are different stages. I'm not sure about around the world, but in my country, we have 3 stages. First is the proposal, which is in 6 months. And then in 2 years, you have your transfer, which is MPhil to PhD, and then your PhD Viva. So if you have written something along the way, it will be easier for you to progress into those stages rather than just taking time and writing things and stopping your research at the same time. So if you're doing a research, you're not going to do that the entire day. So if you focus your experiments for like 4 to 5 hours, just write for 1 hour and then go home. Take a refresh, come back, do your experiments, write 1 hour. If you write 500 words a day, if you multiply that by 20 days a month, for an example, if you just work only 20 days, how much have you completed? You have done almost 10,000. So within a month, you have written 10,000. Even if it's 100 words, it's fine because you have 3 years for your PhD. So make sure you write whatever you have done, and then it will be easier for you at the end of the day.
Perfect. That means keep living the PhD while you're at it from the day one. Right, and we would genuinely like your honest feedback, and we will give you the link for a feedback form in the chat. By all means, let us know what you think. And we have one question in the chat coming to me. Do you have any advice on how anyone could secure a PhD? And at the same time, I think doing a PhD in medical sciences is a bit expensive. So any advice getting on a scholarship?
If you want a fully funded research, just go into, there's a website called Find a PhD. It's very important because you'll find a lot of funded PhDs in there. But if you're capable of funding yourself, so I would say find a good university that you want to work in, and then talk to a person that's like a lecturer or a supervisor that you want to have as your supervisor. Talk to them directly. Don't just be shy or anything. Just email them. Make sure they will get back to you and tell them what you want to do and if they are interested in taking you. So please make sure that you talk to them directly, because if you talk to the university, it won't be enough, because some of the lecturers don't publish the funds that they have. So they might have funds to take you in, but not precisely like for the school fees, but at least to buy the equipment or the cells or antibodies or whatever you want. If they can help you, that would be a good thing. So my suggestion is just go and find on find a PhD on a link, and then find fully funded ones. But currently, it's really hard for international students to get a fully funded PhD unless you're like really good. But if you're in a university, talk to the lecturers, ask if they have any PhDs which are fully funded or at least half funded, which will help you. That's the only thing I can give you, because with sciences, it's a bit expensive because you don't need to buy antibodies, cell lines, reagents, all the bulks. So if you're not fully funded, it's going to be really hard. Luckily, my supervisor had funding from undergraduates and postgraduates. So while I was teaching, I utilized that money for my research at the same time, because my students did something similar to what I have done. So I used my resources for their research, and I utilized their money to buy the rest of my stuff. So you have to work through and you have to talk to your lecturer or supervisor regarding that. That's the most important thing.
Absolutely. So it's find a PhD, and there I think you'll find a lot of PhD scholarship offers in, I think it's a global platform. You can find PhDs all over the world. Yes. And while we talked about supervisors, I've heard that having a good supervisor is sometimes more important than having a good project, because that can change your entire PhD journey. Any tips on finding a good supervisor in there?
Yeah. Luckily, my supervisor was like a second mom to me. Like, even if I don't come to the university for one day, she will call me and ask, "Where are you?" That kind of person she was. So I was very lucky to have all three of my supervisors were like that. So I was really lucky to have my main supervisor like that, and the others supporting her as well. So if you have gone to a university and you know the staff members, and if you know how they are, that is really key. Because if you know that they're very grumpy, they don't like people much, then that's going to be a struggle. But if you know someone who's friendly, who will talk to you, who is willing to help you, then that's really good. Because even if you have an amazing project, as Ravini said, if you don't have a good supervisor, you will go down because you're not motivated enough, because the supervisor is not telling you what to do, because it's your research. You will do it on your own, but they will guide you, and then they will guide you in life as well. So you need that when you're doing research, because you're as I told you in my presentation, you're isolated, you don't have much support, your friends and family are away from you, you're alone, you need that mental help, because without that, your PhD, it's really hard to do a PhD, because I know I have seen people struggling with PhD without the support of their supervisor. So they might have to change to another one, because if you change in between, it will be a struggle, because it's not the same way one person thinks then similar to the other person. So you'll have to change a lot of things. So whatever you're doing and whoever you're going to select as a supervisor, make sure you select the correct person. Talk to them. Once you talk, you know what kind of person they are. So make sure you select the right person.
Absolutely. And your lab mates and your supervisors and the networking, that's really important, because your supervisor is going to be your mentor for a long time.
Yeah. So
yeah.
friends is also another key factor, because if your friends are trying to pull you down, then they are not your friends. They need to keep you motivated. Like, even if you're down, they should be able to help you, make sure you're happy, and then support you throughout the way. Luckily, I had good friends during my PhD who were very helpful, who supported me throughout my PhD. So in a way, you need to select who's the best friend, who's your friends, who's going to be till the end of your life, because some people will just take your support and just leave.
Absolutely. We have one question from Facebook. Yep. It says, I would like to know, what is your sample size and what is your analysis method, whether it's done by statistically or numerically?
I did statistical analysis because I used 50 specimens, tissues, tissue specimens. And then I used Minitab as a statistical software. And I used Spearman's correlation, Kruskal-Wallis test because of my analysis, because I wanted to do the significant correlation between vascular mimicry and different stages and grades to say that it's highly expressed in high grade and higher stages cancer. So there are different softwares you can use. It's in my university, it's mainly SPSS and Minitab, but it can be different from other universities or around the world. You can use any software as long as you have a significant correlation or a significant number to prove your techniques are correct.
Okay, and another question. So you used 3D models for imaging.
Yeah.
Is there any specific steps you took? Because it's not like 2D cell imaging. Did you take any extra steps or any techniques?
Yes, with 3D, you have to be very careful, because if there's a contamination, the whole experiment will fail. So you have to start from the beginning. So the 3D models that I created, I use collagen and fibronectin. So I bought collagen bulk, and then I diluted it, and I used sterile cover slips. So if you get like a 6-well plate, you put sterile cover slips on it, and then you put fibronectin and fibronectin and collagen on it to make the ECM. You incubate it for 1 day, and then you take it out, and then you put the cells on top. So, and then you incubate it until it takes, because 2D, it will grow within a day or two. It depends on the the fast way of reproduction. But with 3D, it will take some time. It will take 3-4 days or 5 days maybe. And if you're doing a normal cell line, it will take more than 2 weeks, because normal cells take time to grow. So make sure that you do it correctly and very aseptic conditions, because if there's nothing, then you'll get a lot of contamination.
Perfect. Are there any questions from the audience? Anyone would like to unmute themselves? Any advice from a registered scientist? Okay, if not, we can wrap up for the day. Thank you very much for being here today. Nirusha, thank you very much. I think it's midday for you. Yeah, so thank you very much for your time. I know it's a busy schedule and you have given us a very good talk, and I think that's really important for anyone who would like to do a PhD and to become a researcher in future. And thank you very much for everyone who attended today. And well, we invite you to join with us for the episode 12 of a PhD Talks by NcodeX on the first Saturday of next month. Thank you very much and have a good day.
Thank you for inviting me. Thank you very much.